Welcome to Vet NeuroJar, the podcast dedicated to exploring the intricate connections between neurology and veterinary medicine. Today, we're diving into a fascinating study that explores the auditory pathways of sheep and its implications for understanding hearing in both animals and humans. The ability to hear is fundamental for communication, environmental awareness, and overall quality of life. When auditory pathways are compromised due to disease, trauma, or other factors, understanding how the auditory system functions becomes crucial. One method used to assess the auditory system objectively is the Brainstem Auditory-Evoked Response, often referred to as BAER. The BAER is an electrophysiological test that measures the electrical activity in the auditory nerve and brainstem in response to sound stimuli. By placing electrodes on the scalp, scientists and clinicians can record how the brain processes auditory information, helping them identify abnormalities in the auditory pathways. This is an important tool in both neurology and audiology, and is used in both human and veterinary medicine. In the world of veterinary medicine, animal models play a crucial role in advancing our knowledge of physiology and disease. Animal models help in the development of new surgical techniques, explore medical devices, and improve our understanding of various conditions affecting animal health. Sheep are increasingly recognized as a valuable large animal model for otological research. Their auditory anatomy shares striking similarities with that of humans. Characteristics of the middle ear, round window, and cochlea are comparable to those found in humans. This makes sheep an excellent choice for studies aimed at improving our understanding of the human auditory system. Sheep are also of a size that allows use of human surgical equipment and implants. Despite the growing popularity of sheep as an animal model, normative data for BAERs in sheep has been lacking. Having access to baseline data is essential for accurate interpretation of BAER results, and this is particularly important when assessing auditory function during clinical trials. A recent study was conducted to address this gap in our knowledge and establish normative values for BAERs in Swiss White Alpine sheep. Let's delve into the specifics of this groundbreaking study. Researchers from the University of Zurich in Switzerland embarked on a retrospective analysis of BAER data collected from 6 female Swiss White Alpine sheep. The sheep ranged in age from 35 to 56 months, with an average age of approximately 42 months, and their weight ranged from 72 to 92 kilograms. The BAER assessments were performed while the sheep were under general anesthesia. The researchers meticulously monitored the sound pressure levels near the eardrum using a probe reference microphone to ensure accurate and consistent acoustic stimulation. Clicks were used as the acoustic stimuli, with stimulation levels beginning at 100 decibels of sound pressure level and were decreased until no BAER signal could be identified. The BAER threshold level was defined as the lowest stimulation level at which wave 5 could be identified. Disposable needle electrodes were carefully positioned on the sheep's head to record the electrical activity generated in response to the auditory stimuli. The electrodes were placed on the ipsilateral retroauricular region, the contralateral retroauricular region, and on the forehead. The electrical impedances of all recording locations were kept below 6 kiloohms. The BAERs were averaged over 2,000 repetitions to improve the signal-to-noise ratio. Now, let's explore the results of the study. The researchers analyzed 15 BAER recordings from the 12 ears of the 6 sheep. They meticulously assessed the latencies, which refers to the time it takes for a wave to appear after the stimulus is presented, and the amplitudes, which refers to the size of the waves, of waves 1, 2, 3, 4, and 5. The study found that the mean BAER threshold, which is the softest sound the sheep could hear, was approximately 45 decibels of sound pressure level. At a high sound pressure level of 100 decibels, the mean latency of wave 5 was approximately 4.35 milliseconds, wave 3 was 2.44 milliseconds, and wave 1 was 0.88 milliseconds. The interpeak latencies, which represent the time intervals between the waves, were also measured. The mean interpeak latency of waves 1 to 3 was 1.56 milliseconds, 3 to 5 was 1.91 milliseconds, and 1 to 5 was 3.47 milliseconds. The mean amplitudes at 100 decibels were 0.04 microvolts for wave 1, 0.5 microvolts for wave 3, and 0.4 microvolts for wave 5. One of the important aspects of this study was the assessment of the reproducibility of BAER measurements. Follow-up measurements were performed in some of the sheep, and the results showed good agreement between measurements taken on different days. The mean absolute latency variation between measurements was small, ranging from 0.12 to 0.18 milliseconds. Similarly, the mean absolute interpeak latency variation was also minimal, ranging from 0.13 to 0.29 milliseconds. This high degree of reproducibility validates the reliability of BAER measurements in sheep and supports the use of these normative values in future studies. The findings of this study have significant implications for the use of sheep as an animal model in otological research. By establishing normative values for BAERs in Swiss White Alpine sheep, researchers now have a valuable tool for assessing auditory function during clinical trials. This enables real-time evaluation of the auditory pathway and provides a reference for comparison when investigating the effects of interventions or treatments on hearing. The study's results offer insights into the auditory processing pathways in sheep and humans. The wave latencies observed in sheep were shorter compared to those reported in humans, suggesting potential differences in the speed of auditory processing. However, the interpeak latencies were comparable between sheep and humans, indicating similarities in the overall timing of auditory events. The researchers acknowledged that the study had some limitations, including a modest sample size. However, they emphasized that the reproducibility of the BAER data was confirmed through repeated recordings, which strengthens the validity of their findings. The researchers also highlight that previous studies on BAERs in sheep have reported varying values, possibly due to differences in breeds, ages, and experimental conditions. Indeed, variability in BAER latencies might be due to differences in breeds, sizes, weights, or anatomical features. Similar studies with dogs indicate relevant effects of maturation, head size, and breed. In the current study, the researchers considered a homogeneous group of sheep to exclude sex, age, and breed dependency of the BAERs. Ensuring accurate acoustic stimulation levels in the ear canal posed a challenge in this study. Because of the narrower and more S-shaped ear canal of sheep compared to humans, the insertion of the earphone can lead to lower acoustic levels. To address this issue, the researchers meticulously monitored the sound pressure levels near the eardrum using a probe reference microphone. In conclusion, this study provides valuable normative data for BAERs in Swiss White Alpine sheep. The establishment of these normative values enhances the utility of sheep as an animal model for otological research and facilitates the investigation of auditory function in both animals and humans. By understanding the intricacies of the auditory system in sheep, scientists and clinicians can develop more effective strategies for diagnosing and treating hearing disorders. The information gained from this study contributes to our broader understanding of auditory processing and paves the way for future advancements in the field of otology. So, let's celebrate the amazing world of veterinary neurology and the dedicated researchers who are expanding our knowledge. Let's appreciate the sheep that have contributed to our understanding of hearing and acknowledge the importance of animal models in advancing medical science. Thank you for joining us on Vet Neuro Jar. Keep those minds inspired, hearts light, and tails wagging.